Arsenic is released into the environment from natural sources, such as the degradation and dissolution of rocks and minerals, volcanic emissions, and from anthropogenic activities like mining, coal and geothermal exploitation, and agricultural practices, as well as industrial and municipal waste, fertilizers, pesticides, paints, and cosmetics. It can contaminate both ground and surface water, as well as soil and air, resulting in human exposure mainly through the intake of contaminated food and drinking water. In fact, the ingestion of contaminated food is the main source for human dietary intake, while non-food contributors such as occupational and iatrogenic sources are mostly limited to single individuals. Inorganic arsenic is primarily metabolized in the liver through two major pathways: reduction and oxidation reactions that interconvert arsenite and arsenate, and methylation reactions that convert arsenite into methylated species. There is large variability in the extent of arsenic methylation between individuals, probably due to a large difference in methyltransferase activity and possible polymorphisms. Arsenic compounds are primarily excreted in the urine as inorganic arsenic, as well as its monomethylated and dimethylated forms. The toxicity of arsenic varies with its chemical structure and oxidation state. Inorganic arsenic is more toxic than organic forms, and trivalent species are more toxic than pentavalent ones. The International Agency for Research on Cancer classifies inorganic arsenic compounds, in particular arsenite, arsenate, and arsenic trioxide, in Group 1, which comprise substances with sufficient evidence of human carcinogenicity. MMA and DMA are classified as Group 2B compounds, which are possibly carcinogenic to humans, while arsenobutane and organic arsenic compounds are in Group 3, as they are not classifiable. Certain groups of people are particularly vulnerable to arsenic exposure, including pregnant women, developing children, and those with underlying genetic risk factors. The margin of exposure of a compound is the ratio between a benchmark dose related to the risk of lung, skin, and bladder cancer and the estimated daily intake. For arsenic, this demonstrates a higher risk of adverse health effects, particularly in infants and young children, because they consume more food relative to their body weight compared to adults. Additionally, they may be particularly susceptible to the adverse neurodevelopmental effects of inorganic arsenic.